Thanks so much for joining us today, everybody. We're pleased to be joined by Dr. Lopez. He grew up in Mexico and attended the National Autonomous University in Mexico, earning his bachelor's in chemistry, followed by a master's in biochemistry, specialty training in electron microscopy, and a PhD in biochemistry. He then came to UCLA as a postdoctoral fellow and now serves as a professor and director of the National Institute of Deafness and Other Communication Disorders National Temporal Bone Lab at UCLA. We're delighted to have him joining us today to speak on current concepts in the biology of otosclerosis.
Okay, good morning. I'm really excited to talk in this grand rounds. For me, I'm just going to give you a brief talk about how I have contact with House Hearing Institute. My first contact was in 1993 with Dr. Linticon, our great scientist that we honored to have him. And then from then, I would continue to work with several investigators at House. And then I was working in basic research in animal models. And in 2000, I began to work with the human tissue. And I focus always my aspect on what happened in the inner ear because of my formation from the chemical point of view. So in this review, I'm going to focus more into what are the molecules involved in otosclerosis, going directly in new concept. Interestingly, in otosclerosis, is the research is kind of sparse here and there. We don't really have a line that follows, like, for example, in hair cell regeneration or even Meniere's, because it's very sparse. So believe me or not, it took me some time to gather research current new information. So in the background, you can see one of the slides that Dr. Linticum uses showing cavitating otosclerosis, and we are going to go over in this presentation. And again, it's a pleasure to me talk to you, and I'm very happy to share a little bit of what we have here. Sorry, I have to just show this quickly. Again, this is our laboratory at UCLA, and you can see I always from a while ago, I begin my talk talking about Dr. Fred Linticum. His contributions to otology are incredible, and I had the opportunity, and this, what you see in the background is my office, and in which Dr. Linticum was spent his last years, and he was really a brilliant person. He was around 95. He asked me, "Ivan, do you think I should retire?" He was already here at UCLA. I say, "No, maybe not." And he was so enthusiastic. He comes early at 7 in the morning, and we begin to work together. I was really lucky to have him and continue and preserve his legacy. So let me remove this. So one of the things that's always important to have into account, especially because I'm talking to otologists, neurotologists, is the human temporal bone research is really, really challenging because, as everybody knows, the temporal bone is the hardest bone in the body. And human temporal research continue to be a challenge to investigate different disorders. So we have a cavern and tissue collection from we can do numerous experiments. It's very exciting. We really got funding from NIH to continue the temporal bone research. And some of these temporal bones have been collected from about 50 years ago. And the nice thing is that they are still useful. We are trying to do molecular biology studies with new techniques, next generation sequencing, and it's amazing that how this tissue is well preserved. So, as you know, the gold standard method for visualizing the pathology of human hearing loss has remained to be postmortem histology, and over for 130 years. It's very useful to determine alteration in the human inner ear. And in our collection, we are very lucky to have tissue stained with patients with eye drops, Meniere's disease, and sudden hearing loss, and otosclerosis. So But first, for all our studies, it's very good to have a very well prepared tissue sections. And we have here, for example, a section from our collection. You can really see that when it's collected in appropriate time, preservation is amazing. You can really appreciate the hair cells, the spiral ganglion area, the stria vascularis, etc. Then we have a database from which you can have all the information necessary to correlate the clinical pathological findings. In our collection, we are very lucky to have around, among other diseases, we have 324 specimens, which may allow us to do numerous research or different type of research in this area. I'm going to share, I'm going to present a little bit the introduction, histopathology, and more focus will be taken in the recent studies, some studies done here at UCLA with Dr. Lintikov and also House, and previous studies with the fellows. So, and I'm directly to the point, and I'm very simple. It's otosclerosis, a progressive temporal bone dysplasia that affects selectively the human otic capsule and is characterized by excessive bone remodeling in the otic capsule. The hallmark of this disease is a stapes fixation with resultant conductive hearing loss. Otosclerosis is the most common cause of conductive hearing loss in adults with normal appearing tympanic membrane. So as I mentioned before, so there's been quite a numerous advances, especially in the past 5 years, about the molecular biology, investigating the genetic changes in the different diseases, among them otosclerosis. And even though this is advances, it has we still don't really know the full details, what is the etiopathogenesis of otosclerosis. And we are going to be presenting recent molecular biology studies and imaging studies that, again, what happened is that these findings have been really new, novel. I mean, I'm talking about maybe 5 years. And it's really what we now, the challenge is to integrate all these concepts, recent concepts, what is prevalent is known. So just briefly a timeline, I'm just going to focus on 1735. Otosclerosis was first described by Valsalva, has ankylosis of the stapes to the margins of the oval window. Then we jump into 1893, in which Politzer discovered the true pathology to be in the labyrinthine capsule. The term otosclerosis has been remaining from then. So etiology, exact causes we're still investigating. Heredity is family history. There is a 50% of the cases. Females are more affected than males, sorry. And the age of onset is usually between 20 and 30 years of age. Pregnancy, otosclerosis might be initiated or aggravated by pregnancy, but never caused by it. Other factors, metabolic disorders and endocrinal disorders. Just a brief classification, probably everybody are aware, but just because of the requirement of teaching, so we put there are clinical otosclerosis and there are stapedial otosclerosis. It causes by stapes fixation and results in conductive deafness. And then we have cochlear otosclerosis that it involves region of around window and may cause sensory neural hearing loss due to liberation of toxic materials. And then we have histological otosclerosis, remains asymptomatic and caused neither by conductive or non-sensory neural hearing loss, but is revealed only at postmortem. And then there are, and I'm going to make just a note that given that this representation recorded, I was very careful and tried to add all the references or books from which I took all the material. So in case you want to review it, if I go superficially, they are always mentioned the source of the reference. So you can go back if you are interested in going further. So among the factors involving the pathogenesis of otosclerosis, we have genetic background involving on different genes, TGF-beta1 or collagen 1A2, then measles virus, so virus have been implicated, some autoimmunity, inflammation, of course, and different type of cytokines involved. and then sex hormones, and there are also environmental factors. So what is really interesting is that there is a molecule called osteoprotegerin. It's an anti-resorptive cytokine, maybe for its ability to protect bone against resorption. So it is expressed in high levels by engineered structure to inhibit OT capsule remodeling. So as you see to the left, in femur, the osteoprogenin is a very low level, so there is constant bone resorption. In contrary, in the OT capsule, the OPG the level is high, and then regulates the rank L or rank system. So there is a very little no bone resorption. So this system is important and could be dysregulated in genetically predisposed individuals, by measles, a virus infection, increased secretion of TNF-alpha-1, interleukin-1 by pregnancy, or hormones like prolactin. This regulation of this pathway system stimulates bone resorption in the OT capsule with the resultant capsular remodeling and possible state fixation. So the OT capsule is that has two main features not found in either the bones of the human skeleton. So there is a very low bone remodeling because of this system I just mentioned before, and contains small regions of immature cartilaginous tissue called globally interossei. So these two unique features might explain the fact that otosclerosis is exclusive to the OT capsule. You can see here a diagram where I really like it because it's very simplified about where is the periosteal layer and the endocardial layer, and the global interossei here, close to the OT capsule, see, and then the endosteal layer. So this interaction maybe makes prone for the otosclerosis. So now about the pathophysiology. So otosclerosis progresses through three well-defined stages. The primary pathological process begins with an inflammatory stage marked by increased vascularity. The second stage is one of active bone resorption, forming a spongiotic bone lesion, otospongiosis. In the third stage, otospongiosis progresses to a final fibrotic healing stage, with the sclerosis characterized by formation of regularly woven, dense and poorly vascularized bone, and caused by aberrant osteoplastic activity. So this is a Chinese stadium showing the different components of otosclerosis. So we have a 60-year-old patient diagnosis with bilateral otosclerosis, and the section is classically Chinese staining, it's a little reddish, but and then you can see really number one, the area of the otospongiotic region. So it's really in contrast with otosclerotic, which is almost like inactive reaction. So it's really remarkable the histological differences. And then, as I mentioned, the otospongiotic region is the active lesion characterized by areas of fibrous tissue containing monocytes, macrophages, and enlarged capillaries, and is easily identified. So you can see here, for example, how large. And then this another characterized that is the alienization of the spiral ligament here in this arrow. And then there is the lateral wall is well preserved in this temporal one. So it's very interesting, and when you study the pathology, and we are lucky to have still this collection of temporal bone sections, you can really see that the organocorti, it's really very hard to see any damage. So we are going to concentrate in this area. And the figure at the bottom, you can see higher magnification view of the otospongiotic portion, shows numerous osteoclasts and osteoblasts in the large marrow spaces that are filled with fibrocytes and blood vessels. You can see really it's very distinctive. So I just, instead of going over and over, because we are more focused into the molecular biolayer, for the students, for the learners, it's very important. This book of Shogness Pathology, and it's really it has a chapter on disorders of bone, and covers otosclerosis very in deep on the different changes in the temporal bone. So I'm just going to recommend to check this book. And this is, we are going to just focus more in the otosclerosis associated with atrophy of the spiral ligament. On this, in this image, you can see really, this is a 75-year-old woman, have a long history of progressive bilateral hearing loss and for a hearing aid in the left ear. Its histological studies showed a large otosclerosis foci involving the lateral walls of the bony labyrinths and foot plates and the stapedies. The view of the basal tone of the left cochlea showed irregular replacement of the osteal layer. You can see here. Of bone and degenerative changes in the spiral ligament consisting of an outer layer of dense connective tissue. Hyalinization, we are going to, and when the next part of the talk, we are going to constantly mention to this phenomenon. Loss of fibrosize and loss of tissue volume. In spite of these changes, all other structure of the cochlea appear normal. So this is what I mentioned. You can really see the organocortex. You see barely any disturbance. And for example, the stria oscularis, but it means that all these changes here are very relevant in cochlear otosclerosis. And again, coming back to Dr. Fred Lintigum and honor him, you can see his contributions. Yeah, about in PubMed, when you look at his name, you have around 239 manuscripts, and from those manuscripts, 49 manuscripts are on otosclerosis. He was he really loved, and he was his passion, otosclerosis. We are lucky. We have a tremendous archive of tissue section with otosclerosis, with different diseases associated. And also going back to it's always good to review what is in the past so we don't repeat it again. And I went back to his first papers, for example, this one that appears, and I'm not saying it's the first one, maybe others, but the one that appeared in PubMed. You can see in this this is an article. It's a current management of hearing loss in children, and he described a lot of diseases and described very well otosclerosis. Then you can see the dates, for example, and I'm going to just mention this paper number 3. They say otosclerosis focus and facial canal. Keleben and Lintigum, they have wonderful papers, and we are going to talk about this labyrinthine otosclerosis. And again, and now go forward from what we were talking 1964, now going back to 2018. And you can see he continues to study the cellular biology of the otosclerosis, which is remarkable. I mean, he went into different proteins like connexin and different proteins. So I'm going to explain you briefly about some of his papers. But you can see he never stopped. And he was so curious, and always among his last wishes was to find out which was the chemicals involved in otosclerosis damage. And just because I really love this image, when I was looking about his manuscripts, I found this paper about cavitating otosclerosis, and it was written. Yes, I mean it's not that old. This is 2010, and then he shows these tremendous empty spaces around the otosclerosis foci. So this is just a correlation with the CT scan, and you can really see the empty space, the cavitating otosclerosis here, and you see the quality of this preparation. And we have to, we are really in the brink of extinction, the temporal bone laboratories, and a little commercial that they're trying to NIH fund our laboratories, 4 laboratories again to continue temporal bone risk, because you can really see this type of preparation. It takes 4 years to have it. I mentioned before that we have it takes like about 1 year to have from the time you collect the temporal bone. But you can see this temporal bone was probably processed 30 years ago. And you can see the qualities. What you have in a temporal bone section is the information is tremendous. And then now I'm going to mention some of the manuscript that Dr. Limpton has about the cellular biological studies and otosclerosis first. So And again, and I'm not going to read the whole thing, but you can see this study about Kellerman analytical is really, really extensive, 68 pages. And then he made, again, he go back to the beginning and you can see the concept of the, I'm just going to read briefly the first sentences. In 1899, Siebenmann formulated his theory according to which metabolites produced by the otosclerosis bone exercise toxic influence and ending of inherent damage. His theory has been adopted by numerous authors down to the very present. No evidence has turned up under the light microscope to substantiate this concept. Results of few chemical investigators searching after the special otosclerotic products possibly remain the only ones to deliver arguments on its favor. So what it means to say is that even from that time and to the day, and you will see at the end of my presentation, in spite of all the molecules that we have detected and genetic backgrounds, so in deep imaging studies, we are still trying to understand what happened. This is just to also given our collection that we have here also from Dr. Rugassen from 1970s, 1980s here at the UCLA. Labyrinthine otosclerosis and sensory was also approached, and you can see here the normal cochlea. And this figure begin to see the changes in the linearization around the spiral ligament. It is very, very pronounced. So this paper was written in 1975. So the problem it was already seen and before of that. One of the papers that it caught my attention because you can see 2004 or 20 years ago, Dr. Lentigo was already directly involved in the molecular or cellular histopathological techniques to understand this problem. So he studied with Johnny Doherty changes in cochlear otosclerosis in the stria vascularis and the effect of hearing levels. And he went into the molecules. Directly, he investigated several proteins that they found out may be involved, they were expressed in the ear. And he was able to detect changes in the different patients. For example, here in A, we have normal cochlea and sodium potassium ATPase is present in the type 2 fibrocytes and the spiral ligament. And then he found that there was some decreased staining around the patient with otosclerosis and in the area of hyalinization. Another, and I apologize, I don't mention other groups just because of the time and because of the familiarity. I just also want to mention one of my teachers, Dr. Raul Hinojosa from the University of Chicago. And about the same time, I remember discussing at house with Dr. Linticon about the hyalinization, and he was talking about the papers that whether or not we should believe that this molecule was involved. And this paper is written by Dr. Hinojosa and also going into the molecular biology. So in spite of being our fathers in otology, they were already involved in the molecular biology. So it was really interesting that nowadays we say that we have all the discoveries again, but they were already into the point, into the problem. So this is just a hematoxylinosin picture from the same manuscript, and you can see the hyalinization. This is the otosclerotic bone and the spiral ligament. And also you begin to use also modern techniques, laser confocal imaging and immunostaining. He tried to identify that he found different molecules here around the hyalinization. And this is more like colocalization, and he could detect all these proteins around, for example, keratin sulfate deposition around the hyalinization area. Ourselves, although not directly studying otosclerosis, I've been dedicating my whole life to painting cells and study what are proteins, or where are located all these proteins in the ear, first in animals and later in human. And we, for example, study sodium potassium ATPase is a very important protein involving homeostasis of the inner ear. This is a normal section of and these are celloidin section. Celloidin was removed and we use antibodies, and you can see really delineate the structure of the inner. This is a normal cochlea. See the organocordia and the spiral ligament and the spiral ganglia. Then you can see when we compare with different type of patients with different disease, among them otosclerosis, it's a remarkable decrease of sodium, potassium, ATPase. I don't see any light up or significant relevance, but this will be the hyalinization area. Now we go to more into the proteins of proteomic studies in otosclerosis. And again, making honor to Dr. Linticon's work. You can see this paper in 2015. He was already within his group, and with all the support he had, interested in what are the proteins present in the inner ear, in the human inner ear, using proteomics. So, and again, I'm making emphasis because for the ones that are familiar, all these techniques of proteomics, genomics, GWAS, etc., has been put to work in other organs or system or the brain, for example, from more than 20 years. The inner is always going behind, and we have been jumping into the different techniques very recently. And in 2015, these techniques were not really the way right now, 10 years later. But he already was working in this methodology. So he was able to detect different proteins in this type of technique. You extract the proteins from a single section of celloidin, and then you submit to a spectroscopy, and then you do some analysis, bioinformatic analysis to detect the different proteins. So he just to show you an example, I don't want to focus on a single molecule, but for example, he found different molecules. And one of what was very, very interesting is TGF-beta, transforming growth factor beta 1 is always highlighting in different diseases or aspects into the inner ear. And then, of course, immunostaining, and then he did identify different proteins, TGF-beta, for example, superoxidase mutase involving oxidative stress, and so on and so forth. We, and recently, 10 years later, and we make a note on Dr. Linticon in honor of his work with Sarah Hodge, and I want to make also a commercial that we've been lucky to have several manuscripts with fellows on house, and hopefully we continue this tradition. We are really, really lucky to have be able to interact with you, and it results in really productive publication, and it really I have had remarkable fellows that really get 2 or 3 publications, and I encourage you to keep coming here. This is your house, and then we continue the research. So this paper is very recent, in which we, based on the proteins that Dr. Linticon detected, we select some proteins maybe involved in otosclerosis. We select different patients, normal and with bilateral otosclerosis, and again, using new imaging techniques, we were able to use a tile scan of the whole section. In the past, observations were done just by looking at small area and going back. It was really tedious and very inaccurate. Now we are able to have the whole image in a single section, and zoom in, and here you can see with different proteins, just to know nitrogen and so on and so forth. And you can see really the otosclerotic region and the otospongiotic region very active with these proteins. You can see also, we can now able to see, apply the polarized filter together with this image. You can see the woven bone, and we are trying to generate one in 3-dimensional images about how is the path of organization of the woven bone in this material. This is a higher magnification, which you can really see. In the number 1, you can see the otosclerotic region, less active, and of course the otospongiotic region, which they're spreading a lot of proteins. And here is pretty much a similar image. So in a protein that he was very particularly interested in, and I knew about this protein because of him, and he was really brilliant. It was a ubiquitin. It's a protein that how is it so complex, the interaction, the ubiquity in the proteins in charge of removing the old proteins. And he said, Ivan, why you don't try this staining? But don't try, and we found this protein also. And then, of course, TGF-beta, which has been implicated in otosclerosis, we stained and we found it in the area of the autospongiotic region. Also applying more fancy techniques, immunofluorescence, confocal, we corroborate our findings using different antibodies and fluorophores. So that is another paper, and again, coming back into the commercial, you can see we have also Mia Miller from House, and then Dr. Linticon pretty much designed this the paper with his idea. Why you don't test, see the connection which is involving different mutations, for example, and hearing loss, see what happened in otosclerosis. So the hypothesis was the connexin 26 is diminishing the spiral ligament in subjects with hearing loss and cochlear otosclerosis. So, and again, using the material that we have in our archival tissue, we select normal, and you can see the expression of the connexin 26 and different proteins, different locations. And when you stain or when you use the same antibody with in a cochlear otosclerosis specimen, you see really a decrease in connexin. So cause effect, we don't know, but there is some effect also in this protein. Recently, we have a manuscript in which we studied the expression of TGF-beta1 and CTGF in the implanted cochlea and in its implication in new tissue formation, and we use some otosclerotic tissue. So again, the relevance is at the top, you can see a cross-section of the cochlea, close to the mid-modulus, not exactly, but you can see the otosclerotic region. This is the path of the electrode. And then you can see H&E, the new bone formation in this area, when this is a different story. But when you detect this protein using the antibodies against TGF, you can see really very active participation, not only around the cochlear, the electrode, and this you can see in green, this TGF-beta, but also around the bone. So making this really a complex issue is you have new bone formation inside of the temporal bone with cochlear implant, and at the same time you have the participation of otosclerosis. So really how to connect all these dots is really big, the challenge for the next years. You can see really the expression around the electro. You can see there is interaction of the CTGF and TGF-beta. All these molecules are related. And always present in all this kind of injuries, so and inflammation and otosclerosis. And we just have and because of the consideration of the time, we just find that, for example, when you insert the electrode, there is electrical stimulation that can stimulate TGF-beta1, and then into generate fibrosis and new bone formation. This is for the electrode. Whether or not this is a similar mechanism or some related mechanism about TGF-beta in otosclerosis remains to be determined. The next part, I'm going to just briefly mention some of the manuscript that I mean, as I mentioned to you, if you can see the dates, really recent, and it's not easy to assimilate so much information, but they are now using now more advanced proteomics. They are able to diagnose different or detect different proteins in patients with hearing loss. For example, using the Perelin, which is an alternative. And they detect different proteins that are highly abundant. They compare, for example, in large vestibular aqueduct vectors, Meniere's, and otosclerosis. And these are some of the proteins. Again, don't worry about what is the protein and so on, but for different iteration in bioinformatics, they came up with proteins that are highly expressed in otosclerosis and also in Meniere's here. They also make comparisons, and you can see, for example, there are other very specific proteins on the otosclerosis. What does it mean? Where are they located? And again, this is really recent. We are assimilating how we can integrate all this concept. And again, now using bioinformatics, which is another area that maybe, I mean, for us, all school people, we need to really go back and do the school. But for the new students, they are more familiar, in which they get all the information generated from other manuscripts from themselves, and they begin to analyze it, how they make other processes are present in very, very tiny amount of tissue or fluid, and then you can detect the protein. So you can see here, for example, yes, and I'm just mentioned that you can see the tremendous amount of proteins. Unfortunately, here we don't have really much highlighted in the temporal bone, in the area of the bone. So again, how you can connect all these dots and how you can interact all these proteins remains to be done. And I'm pretty sure with all this new methodology of bioinformatics, we will be able to integrate how these proteins interact with the bone. And then you probably are familiar with the term in silico, in which the previous studies, they compare all this. They made like a meta-analysis of all the proteins expressed, and they found how some proteins may be more present in Meniere's, some proteins may be more present in otosclerosis, and so on and so forth. So this here, I'll just put the arrows. You can see that, for example, they have all the vestibular schwannoma, Meniere's, and last vestibular aqueduct, the sensory neural hearing loss, otosclerosis, and every protein that they detected with different by statistical analysis and level of expression. These are some of the proteins that they detect in Meniere disease. Again, maybe doesn't make any sense, but as it means we need to do like more in-depth analysis, how these proteins are, where are they, and how they express in which pathway. So just to put you in context. Now, just briefly, we are going to go into what are they in terms of, and also just to mention the manuscript, I didn't two part, but because they are present, and you can go over in the genomic studies and the RNA studies. And this is just one example. And again, if you see the year that when these papers were made, really, really recent, and when they take another type of, a phenotype of first otosclerotic families linked to autos 10. So I have about otosclerosis 1 to 10, and then they found a recently one another mutation. And then they put all the different type of phenotypes and the different otosclerosis loci. So it's able to see all these changes on the different type of comparing with the audiometric data. Animal models is another limitation that we have, and the human ear is, even though there are tremendous amount of investment using mice, other species for injury research. For example, and you are aware, and Meniere's disease, for example, they're really, really hard to replicate or to obtain an animal model. And in time also, for example, an animal model for otosclerosis, I just found one manuscript, and it's recent also, in which they find a mutation in one protein. It's called SMARCA4, and causes similar changes in the mouse. So it's really, and I haven't gone really in deep on the manuscript, but just to mention to you that the lack of animal models we have here for otosclerosis. Genome-wide screens are tremendous now, and again, and I'm apologize for being repetitive, but you can see really this information is really, really making, and you can see the amount of people needed to do this time analysis. And they detect them using genome-wide screen of otosclerosis, using a population biobanks, 27 loci, and some of these shared with the skeletal structure. So, and I don't put the paper, but you can see, and they report this is the largest US of otosclerosis, including a total of 3,504 cases. And they compare with, you just see the number, almost 1 million of controls. So it's really tremendous, and different banks together. And I'm a little biased, but you can see that among these reporting genes, it highlights TGF-beta1, another protein called MEPE. And then using the meta-analysis and then putting them together. So I think this is just tremendous amount of computer time, but you can see really highlights again. There are other molecules, of course, but which one highlight TGF beta 1 and SMAD3, for example, collagen, proteins that are present and keep appearing over and over with different methods, etc. And this is just histology in which this is the capsule, and this is they just tested these two proteins in the mouse, and then they found from X, for example, found in the capsule and maybe around the spiral ligament. So who knows? Maybe this is the molecule that we are looking for. And then also other techniques, and you are familiar with the students here present this paper. For example, they use also perilines to study microRNA in different types of otosclerosis. They did detect different type of proteins also. Collagen 1A1, for example, collagen always highlights, kind of understandable. But the limitation, and they pointed out, is that are really useful to use the perilines to study MRA interference. But however, they could not find really conclusive results because of the amount of fluid they collect is really, really limited. So they still doing an ongoing analysis. And just at the end, just to not step aside, we have also, there are contemporary images techniques. And this is just to mention to you. And again, go intern, though it will take hours forever, but just to have you an idea. For example, they use analyze otosclerosis on their micro CT, and they find new signs using these techniques. And you can really see this is a micro CT raw image of the cochlea. You can see really, it's almost like a histological section. I mean, you can appreciate, and then using different types of imaging techniques, serial section imaging, stacking, and segmentation. They claim that they can see changes inside of the cochlea, not just around the optic capsule, which is really remarkable, because if this is the case and they can detect changes in a different situation, that you can really, and one day I presume it's going to be possible to see in people all these changes. And then finally, and we have a technique called synchrotron phase contrast images using for otosclerosis, and this is the group of Dr. Agrawal, Sumit Agrawal in Canada, and we have some collaboration with them. But in this case, what they did here is how you use this imaging technique. It's a synchrotron. It's almost the size of a particle accelerator. It's really huge machine that is very, very complicated to use. But what happens is that you can get images without cutting the section, without cutting the tissue, and you can see the product, the one they scan, and the tremendous amount of computer time. And then you do segmentation, and you can identify, for example, here in brown is all the vasculature of the cochlea. This is a normal one, and all the structure. And it's really, really remarkable how much information. And again, the important thing is this tissue is not sectioned. So what we are doing is send it to him. He does cyclotron imaging, and then he sends us back, and we do the histology, and we're trying to corroborate or correlate the histology with the synchrotron technique. But in this case, they did this study with the otosclerosis, and you can see in green the area of the damage of otosclerosis in using these techniques. Again, these are really, really manuscript that just emerged around this year or past year. So it really, really will take some time. Go back there and try to translate our 2D image visualization with the clinical and with the three-dimensional image visualization. So this is one image, and this is no CT. This is how it looks. Although the resolution is still far from going to look into what happened, and they will achieve what happened to the hair cells or spinal ganglia neurons, but they give you really, really a glance of what happened with otosclerosis. And they do this different reconstruction. And again, when we talk to them, they use supercomputers, 2 or 3 or 5 computers aligned together to recreate these images. So you can see the area of otosclerosis here. Very, very well delineated. And these images are just superb. Really, really help you. And For example, for me to going from the 2D and translate it into the 3D and then later 4D, you can really have an idea. How's the process? Can we really understand the time course of this disease and others? I think we will in few years. This is just a model showing the different process we already discussed. And just as a conclusion is that, and again, I'm going back into the molecule. For cochlear otosclerosis, the molecular substrate remains to be determined. And again, I'm betting, and there is a likely candidate that appears to be constantly both beta 1. How it works, how interact, listen, I mean, we are still trying to dissect these pathways, how we connect it. And as I've been mentioned constantly, the integration of all the methodologies, and it will take several persons or experts in bioinformatics and so on and so forth, together with the clinical data constantly generated, will really help us to understand how not only otosclerosis but other diseases affect the ear. And this, of course, will be the well benefit for everybody, especially for the patients, in which we really put all our effort to create the best for them. And we have having all this interaction with the basic researchers and otologists to understand all the histopathology and the molecular biology of pathology. And I guess with this, I'm gonna remove my screen, and I really thank you so very much for your time.